The second method we're going to look at is FACS, or fluorescence activated cell sorting. This is a form of flow cytometry which uses light scattering to characterize the property of cells and then sorts those cells based on the properties seen in light scattering. So, in flow cytometry, cells are first stained. In this case, they are fluorescently stained before being injected into the machine. While in the machine, ultrasonic vibrations will then aid in droplet formation, and the system is set up in a way so that each droplet will only contain one cell. So as those droplets are passed in front of the laser, you're only analyzing one cell at a time. Depending on the characteristics of those cells, different wavelengths of light will be emitted, and those will tell the story of the properties of the cell. Now, specific to FACS, after the fluorescence activity of the cell is measured, different charge groups will be attached to each droplet depending on their fluorescence readout. In the case of this study, soluble wild-type ACE2 was incubated with cells expressing the wild-type spike protein of SARS-CoV-2 or the library of the deep mutagenesis scan. This is the scan that mutates each individual amino acid one at a time. So that is what was the cells that were suspended in these droplets. And then they were sorted based on their binding ability and the characteristics of that binding. So as they're run through flow cytometry, they're independently analyzed to correlate the level of soluble ACE2 binding with spike protein expression. Then through the fluorescence sorting activity, the highest and lowest binding 20% of the mutation library expressing cells were then grouped and rerun through FACS to sort into high and low binding groups and understand the difference between the two. Here are the flow cytometry FACS readouts. In section A, we have the soluble ACE2 wild-type receptor being incubated with cells expressing the full-length wild-type spike protein. As opposed to section B, we have the same receptor, but these cells were incubated with the saturation mutagenesis variants of the receptor binding domains. Between those two charts, you can see that in section B, the RBD variants had a larger spread in terms of their binding capabilities to soluble ACE2 wild type. Moreover, you can see more dots for cells expressing high soluble ACE2 wild type binding, as well as more dots lower in the chart showing lower wild type binding, so larger variance when it came to the different mutants. And then in C, you have the FACS sorting, being able to sort the highest and lowest 20% of cells when it came to their binding capabilities into two different groups, which were called the ACE2 high sort and the ACE2 low sort. In light of the results of these FACS readouts, the researchers also wanted to understand which commonalities exist between high sort and low sort cells and which amino acid changes could have influenced these differences. So here they employed enrichment ratios to quantify the prevalence of each mutation in each of the high and low sort cells. From this, they found that in general, RBDs that were expressed shared a highly conserved hydrophobic core, which is clearly depicted in figure A by these blue amino acids toward the center of the figure. The residues surrounding the core that are exposed to the surface were much more variable and mutationally tolerant, which is shown by the different colors surrounding that hydrophobic core in figure A. In high sort binding RBDs, which is shown in figure C, the exposed surface was slightly more conserved but still retained similar amounts of mutational tolerance. These findings of a highly conserved RBD core but a variable and mutationally tolerant exposed interface mirrors the findings that we previously talked about with the 7 beta coronaviruses that were amino acid sequenced and analyzed and given conservation scores. So in light of these findings, we have to ask what's next. We know that successfully expressed spike proteins contain a highly conserved hydrophobic core, but a variable exposed surface that's left to interface with the ACE2 receptor. In light of this, cells expressing high sort RBDs are slightly more consistent in exposed surface makeup when compared to the general population of RBD mutants. So, is there a way to exploit these mutations to target viral RBDs to bind to a decoy? As we saw in the BLI-derived KD values, soluble decoy receptors bound stronger to all 7 beta coronaviruses that were analyzed as compared to the wild-type ACE2. However, are there escape mutations that may occur that will lead to decoy recognition and preferential wild-type binding by the virus? Now we'll take a more granular view of the decoy design to understand which findings led to this specific construction. So researchers noted that mutations at certain positions, particularly between residues 480 and 505 of the viral spike protein, that were favorable for ACE2 interaction. Some of these trends included, for example, position 505 showed increased conservation as compared to other exposed residues. Additionally, residue 489 showed a preference for aromatic residues, and residue 485 was widely tolerant to many polar amino acid substitutions. Additionally, aromatic amino acid preference at position 501 was also noted, which we discussed in the case of the N501Y mutation in last week's paper. So the researchers engineered a decoy that capitalized off of these trends and employed certain mutations at the interface of ACE2 and the viral RBD that would increase interaction. So this took form in aromatic substitutions at positions 27 and 330, and as well as polar substitutions to residue 79. Each of these substitutions were made on the receptor as opposed to on the RBD. So I've highlighted these positions with boxes in the right two figures, and figure A shows each mutation in the context of the ACE2 interface, which is shown in the green at the bottom of figure A. Also, figure A depicts other areas of mutation within the RBD, the viral RBD, that increase specificity to either the decoy or the wild type receptor. So these can be seen in the top figure of figure A. The pink areas denote where mutation led to increased specificity for the decoy, while blue denotes areas where mutation increased specificity for the wild type receptor. So what the researchers did is they pitted the decoy against the wild type receptor again. This time they incubated the wild type spike protein with competing populations of both the soluble wild type and the soluble decoy receptor. This is shown in figure A. In figure B, cells expressing the deep mutagenesis scan of RBD variants were incubated with the same mixture of competing decoy and wild type receptors. It is in this graph that you see it's more apparent that a subgroup of mutants is appearing that seem to preferentially bind the decoy receptor as opposed to the wild type receptor, which is shown by these dots rising above the apparent trend line in the middle of the graph. Then, using FACS, the top 20% of cells binding either the decoy or the wild type receptor respectively were grouped, showing that binding between the decoy receptor and the top 20% of cells that it bound was higher in affinity than that of the wild type receptor. This is shown by the prevalence of points in the magenta box as opposed to the green box. So in other words, a population of mutants showed preferential binding to the decoy receptor, while the subset of mutants that bound preferentially to the wild type was significantly smaller. In further analyzing these mutants, researchers found an increased preference for the decoy receptor was correlated with a hotspot of mutations, one of which was noted at the periphery of the engineered ACE2, which was to be expected, but as well, one was noted between amino acids 498 and 506. So this section between 498 and 506 is specific to a part of the RBD that is specific to a beta hairpin motif that then packs against a collection of alpha helices on the ACE2 interface during binding. No such mutational hotspots were found, however, with regard to wild-type binding preferences. But researchers still were interested in that small group of variants that showed increased preference for the wild-type receptor and wanted to see if this was just noise seen on the machine or if this was truly mutants to be worried about. 24 mutants that were enriched in the preferential wild-type binding sort, which you can see are represented by the blue dots in figure B, were then tested again by a FACS. In the upper right-hand corner, you can see two examples of the FACS results that were done for each mutation. After excluding RBDs that did not bind either receptor, you can see that no dramatic trend appears in terms of subpopulations that seem to preferentially bind one of the receptors over the other. And this trend was seen consistently throughout each mutation. However, they chose to investigate two mutants even further. Those were, interestingly enough, aromatic substitutions to amino acid 501. This was likely due to the emergence of mutations such as 501Y that was occurring in the UK at this time, and they looked into those further. Flow cytometry of those mutants showed, which is in figure C, that these mutants did show an increase in preference for wild-type receptor. However, the binding was still strongest to the decoy receptor. So this binding strength is illustrated by the solid lines in figure C and how they consistently rise above that of the dashed lines, which correlate to wild-type receptor binding.